Before we delve into the sophisticated sphere of healthcare, let's take a moment to acknowledge the basic principles. The heart of living processes, the fulcrum of all biological systems, is the central dogma of life. This chief principle sets the stage for everything we are about to explore. A simple yet profound concept. DNA, the blueprint of life, is transcribed into RNA, then translated into proteins, the cellular engines. This flow of information, from DNA to RNA to protein, fuels life as we understand it. Delving deeper into the proteins, which are the cellular engines, we find enzymes, specialized proteins that speed up biochemical reactions. The principles of enzyme kinetics play a crucial here. It is the study of the rates at which these enzymes catalyze chemical reactions and how different factors, such as temperature and substrate concentration, can influence these rates. Understanding these principles is crucial to manipulate biological processes and even to create new ones. But what if we could modify these processes? What if we could control the speed, efficiency, precision of these enzymes to design biological systems that function like well-oiled machines or computers? This is where synthetic biology and metabolic engineering come into play. Metabolic engineering is a subfield of synthetic biology that specifically focuses on the design and modification of cellular metabolic pathways. It's about reprogramming a cell or organism to improve the production of a specific substance or even produce an entirely new one. Visualize coding a computer, tweaking its operations, and even creating new organisms. That's the power of biology. It has the potential to not just revolutionize healthcare, but also fields like agriculture, energy, and environmental stewardship. Imagine possibilities if we could custom design organisms to produce biofuels, clean up toxic waste, or even fight diseases. With this groundwork laid, we are now ready to delve into the marvels of cellular and molecular biology, genetics and genomics, microbiology and biochemistry, fields that are persistently pushing the boundaries of what we deemed possible. So, buckle up as we navigate you through the evolution of these scientific concepts in healthcare. Having grasped the central dogma of life and synthetic biology, let's now unravel the role of scaffolds in bioengineering. Picture a scaffold, a structure that provides support and guidance. In the world of bioengineering, scaffolds play a similar role, but at a microscale level. They are structures that provide a template for cell growth and tissue formation, giving cells a framework where they can multiply and differentiate. These scaffolds mimic the extracellular matrix, a complex network of proteins and carbohydrates that surround cells in our bodies. This matrix provides cells with a natural environment, fostering their growth and differentiation. By replicating this matrix, scaffolds create an environment that encourages cells to behave as they would in the body. The application of these scaffolds is especially significant in the field of tissue engineering and regenerative medicine. Here, they serve as a support system for cells, aiding in the repair and replacement of damaged tissues. Imagine an injured knee or a damaged heart muscle. Scaffolds can be used to grow new cartilage or heart muscle cells, which can then be implanted into the patient, helping them heal in a way that was not possible before. Scaffolds, therefore, play a crucial role in bioengineering. They provide a platform for the creation of functional tissues and organs, pushing the boundaries of what's achievable in healthcare. Their role is not just confined to regenerative medicine, but also extends to creating in vitro models for drug testing, studying disease progression, and much more. As we continue our journey into the depths of bioengineering, keep in mind the significance of these unsung heroes, the scaffolds, the silent enablers of life. Right after setting the stage with the central dogma of life and synthetic biology, let's delve into the fascinating world of biomedical imaging. This discipline sits at the crossroads of biology, physics, and medicine. It's a crucial tool in bioengineering and healthcare, providing a window into the body that allows us to diagnose diseases, monitor treatment, and understand complex biological processes. We begin at the cellular level, where imaging techniques like fluorescence microscopy, confocal microscopy, and electron microscopy allow us to visualize cells and their components. These techniques provide detailed images of cellular structures, from the nucleus to the mitochondria, and even allow us to track the movement of molecules within cells. Imagine being able to watch as a cell divides, a virus invades, or an enzyme catalyzes a reaction. Such insights fuel our understanding of biological systems and pave the way for new treatments and therapies But biomedical imaging isn't just about visualizing cells, it's also about seeing inside the body without the need for invasive procedures. This is where medical imaging techniques like ultrasound, computed tomography, CT, and magnetic resonance imaging, MRI, come into play. Ultrasound uses sound waves to produce images of soft tissues like muscles and blood vessels. CT uses X-rays to create detailed cross-sectional images of the body. MRI leverages the magnetic properties of certain atomic nuclei to provide detailed images of the body, especially soft tissues. These imaging techniques are invaluable in diagnosing diseases, from cancer to heart disease, and in monitoring treatment progress. They provide physicians with a detailed view of the body's interior, guiding surgical procedures and making therapies more precise and effective. Biomedical imaging is more than just taking pictures of the body. It's about understanding the body's structure and function at various scales, from the molecular to the whole body level. It's a window into the body, offering insights that drive innovation in healthcare and bioengineering. As we continue our journey, keep in mind the crucial role that biomedical imaging plays in shaping the future of healthcare. Before we venture into the realm of bioengineering, it's essential to appreciate the mechanical properties of biological tissues and key biological systems. Biological tissues, from muscle to bone, are not just passive structures. They are complex materials with unique mechanical properties that influence their function. Consider our bones. They're not just rigid structures providing support, they are dynamic tissues, continually reshaping and adapting to environmental stresses. Or take our muscles, they can contract and generate force, yet they remain flexible and resilient. Each tissue in our body, from the heart to the skin, exhibits an exquisite balance between strength, flexibility, and resilience. Understanding these properties forms the cornerstone of bioengineering, but tissues don't function in isolation. They work together in complex systems, each with its unique mechanical properties. The cardiovascular system, the musculoskeletal system, and the nervous system are prime examples. The heart's rhythmic pumping, the coordinated movement of muscles and bones, the rapid transmission of electrical signals in the nervous system. All these are intricate dances of mechanical processes. Understanding these systems and their mechanical properties is the foundation of bioengineering. It's like studying the laws of physics before building a skyscraper or learning the principles of aerodynamics before designing an airplane. By comprehending these properties, bioengineers can design solutions that work in harmony with our bodies, leading to more effective treatments and therapies. This understanding is the stepping stone to the world of bioengineering, where we harness these mechanical properties to revolutionize healthcare and beyond. Welcome to the intriguing universe of bioengineering. The principles of biochemical processes, enzyme kinetics, and metabolic engineering lay the groundwork for this remarkable field. But what exactly is bioengineering? It's a diverse field that fuses engineering concepts with biology to create innovative solutions for real-world challenges. The history of this amalgamation of biology and engineering can be traced back to the understanding of DNA, the blueprint of life. The mastery of this blueprint through manipulation has led to the emergence of genetic engineering, a forerunner of modern bioengineering. Over time, bioengineering has grown, opening new spheres like synthetic biology, which we will delve into. But the realm expands much further, about gene editing technologies, a prime accomplishment of bioengineering with the potential to reprogram the code of life. We'll explore one such technology, CRISPR, later in this video. Additionally, there's bioprinting, a technology that's actualizing science fiction by fabricating three-dimensional structures of living tissue. Together, these advancements are transforming healthcare, ranging from diagnostics and treatments to the synthesis of artificial organs. And this is just the tip of the iceberg. As we journey deeper into the world of bioengineering, we'll discover more wonders set to redefine our future. So, prepare yourself as we continue our exploration into the alluring universe of bioengineering. Have you ever wondered how the future of healthcare might look like? What role will bioengineering play in that future? A world where biology and technology intertwine, creating a tapestry of innovation that propels us towards a healthier future. This is the world of bioengineering, a field on the precipice of transforming healthcare as we know it. Bioengineering is the application of principles of biology and the tools of engineering to create usable, tangible products. It's like a bridge, a connection between the complexity of biological systems and the practicality of engineering solutions. This fusion is leading to a revolution in healthcare, generating novel approaches to diagnosing, managing, and treating diseases. The microscopic to the macroscopic, bioengineering has the potential to change it all. Picture nanoparticles, tiny particles that can deliver drugs directly to cancer cells, sparing the healthy ones. Now envision prosthetics that respond to nerve signals, allowing amputees to regain their mobility and independence. These are not mere figments of our imagination, but realities made possible by bioengineering. But we're not stopping there. We're pushing the boundaries of what's possible. We're talking about gene editing technologies like CRISPR, which could potentially cure genetic diseases. Imagine a world where conditions like cystic fibrosis or sickle cell anemia are a thing of the past. That's the power of bioengineering. And then there's the realm of artificial organs. Instead of waiting for organ donations, what if we could grow a heart or a kidney in a lab? Bioengineered organs could be the key to saving countless lives. This video will venture into the fascinating world of bioengineering. We'll explore the dawn of this field, the rise of CRISPR, the age of artificial organs, and what the future might hold. We'll uncover the marvels of bioengineering and how they're reshaping the future of healthcare. Let's embark on a journey through time and witness the evolution of bioengineering in healthcare. The story of bioengineering begins in the late 20th century with the advent of genetic engineering. In the heart of the 20th century, scientists managed to unravel the enigmatic structure of DNA, the very fabric of life. This discovery was the equivalent of finding a blueprint to life, a roadmap that could lead us to understand how organisms function, grow, and evolve. As we delved into the world of DNA, the birth of genetic engineering was inevitable. The 1970s brought about the first successful experiments in genetic engineering. Scientists were now able to take a piece of DNA from one organism and insert it into another. This was a groundbreaking achievement. Imagine being able to take the best traits of one organism and bestowing them upon another. It was like playing God, but with a pipette and petri dish. Suddenly, the possibilities seemed endless. We could potentially engineer crops to be more resistant to pests, or create bacteria that could produce insulin for diabetic patients. This was not just science fiction anymore, it was becoming reality. But with great power comes great responsibility. As we began to manipulate the very building blocks of life, ethical questions began to surface. What are the implications of altering an organism's genetic makeup? Could we inadvertently create a new disease or a superbug resistant to all known antibiotics? These were the questions that the scientific community and society as a whole had to grapple with. As we moved into the 21st century, genetic engineering continued to evolve. New techniques were developed, each more precise and efficient than the last. We're now able to edit genes with remarkable accuracy, a feat that was once thought to be impossible. The early days of bioengineering were filled with excitement, trepidation, and a sense of limitless potential. We had unlocked a new realm of possibilities, a new way to understand and interact with the world around us. This was just the beginning, the first step towards a revolution in healthcare. Fast forward to the 21st century and we witnessed the rise of a new technology that could change everything: CRISPR. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, is a revolutionary tool in bioengineering. It's a bit like molecular scissors, allowing scientists to modify an organism's DNA with an unprecedented level of precision. It works by guiding a protein, called Cas9, to a specific location in the genome. Once there, the protein acts as a pair of molecular scissors, snipping the DNA at that precise location. Scientists can then add, delete, or replace the genes at this point, effectively reprogramming the genetic code. The implications of this technology are far-reaching, especially in healthcare. Imagine a world where genetic diseases are not a life sentence but a curable condition. With CRISPR, we could potentially rewrite the genetic code of individuals suffering from diseases like cystic fibrosis or Huntington's disease, effectively curing them. And it's not just genetic diseases that could be targeted. CRISPR could also be a game changer in the fight against cancer. By modifying the genes of immune cells, we could potentially engineer them to recognize and destroy cancer cells more effectively. But like all powerful tools, CRISPR comes with its own set of challenges. The ethics of gene editing are complicated and still largely uncharted. There's a fine line between curing diseases and potentially altering the course of human evolution. And while CRISPR is incredibly precise, it's not perfect. Unintended changes to the genome, known as off-target effects, could have unforeseen consequences. Despite these challenges, the potential of CRISPR is undeniable. It's ushering in a new era of personalized medicine, where treatments are tailored to an individual's unique genetic makeup. It's an exciting time to be alive, as we watch the boundaries of what's possible in healthcare rapidly expand. The potential of CRISPR is enormous, but it's just one piece of the bioengineering puzzle. Imagine a world where organ transplants are no longer needed, where we can create artificial organs in a lab. That world is closer than you might think. In the realm of bioengineering, the creation of artificial organs is a marvel that's transforming the medical landscape. It's an exciting fusion of biology and technology, a testament to human ingenuity, and it's happening right now, from lab-grown tissues to three-dimensional printed organs. Let's take a step back for a moment. Picture this. You're in a lab where scientists are cultivating cells, coaxing them to replicate and form tissue structures. These aren't just any cells, they're stem cells, the body's master cells, with the potential to become any type of cell. By guiding their development, scientists can grow specific types of tissues. It's a painstaking process, but the result? Lab-grown organs tailor-made for patients in need. Now let's leap into the future. Picture a different lab, this time equipped with a three-dimensional printer. But this isn't your ordinary printer. It's a bioprinter, capable of printing layers upon layers of cells to construct an organ. This is not science fiction. It's a reality we are inching towards each day. So what does this mean for the future of healthcare? Just envision a world where organ waitlists are a thing of the past. A world where the risk of organ rejection is significantly reduced, if not completely eliminated. A world where patients in need of an organ transplant can have one created specifically for them, using their own cells. The potential to save countless lives is immense, but it's not just about saving lives. It's about improving the quality of life for millions of people. It's about giving hope to those who had none. Yet it's important to remember that we are still in the early stages of this bioengineering revolution. There are challenges to overcome, ethical considerations to address, but the potential benefits, they are simply too significant to ignore. We stand on the brink of a new era where bioengineering could redefine the very nature of healthcare. So, what does the future hold for bioengineering in healthcare? Imagine a world where diseases are not just treated, but prevented before birth. This might sound like a science fiction, but with advancements in bioengineering, it's becoming less of a fantasy and more of a reality. The future of bioengineering is brimming with possibilities, from gene editing to prevent diseases to bioengineered organs for transplants. Picture this. A pregnant mother walks into a clinic. The doctor analyzes the genes of the unborn child, identifies a potential genetic disorder, and corrects it using CRISPR technology. The child is born healthy, free from a disease that may have plagued them throughout their life. This is not a far-off dream. Scientists are already working on these techniques, and successes in the lab indicate a promising future. Now let's take another scenario. A man is suffering from liver failure, and he's been on the organ donor list for years. Instead of waiting for a compatible donor, imagine if we could grow a new liver in the lab, tailored specifically for him. Bioengineered organs, grown from the patient's own cells, could eliminate the need for organ donors, drastically reducing waiting times and the risk of organ rejection. But with these advancements come challenges and ethical considerations. Gene editing, for instance, raises questions about the morality of altering human genes. Could it lead to so-called designer babies, where parents pick and choose their child's traits? And what about the cost of these treatments? Will they be accessible to everyone, or only to those who can afford them? As we move forward, we need to balance the incredible potential of these technologies with ethical and societal implications. But one thing is clear: the future of healthcare lies in bioengineering. It's a future where diseases are prevented before they even start, where organ shortages are a thing of the past. It's a future that's both exciting and full of potential. The future of healthcare lies in bioengineering, and it's a future that's both exciting and full of potential. Let's delve further into the magic of 3D printing and bioengineering. 3D printing, a process of making three-dimensional solid objects from a digital file, is opening up new frontiers in bioengineering. Imagine a world where patient-specific models are created for surgical planning, making procedures safer and more effective. Now picture a world where prosthetics are not just generic, but custom-built to fit the individual perfectly. That's the potential of 3D printing and bioengineering. But it doesn't stop there. Scientists are now exploring the realm of bioprinting, a technique that could one day allow us to print human organs. Yes, you heard it right, printing organs. It might sound like science fiction, but the reality is closer than you might think. Bioengineers are already making strides in printing tissues and small-scale organ models. The next big step? Printing fully functional organs that can be transplanted into a human body. The future of 3D printing in bioengineering has the potential to revolutionize healthcare by making personalized medicine a reality. This means treatments and therapies that are tailored to the individual, drastically improving their effectiveness and reducing side effects. The magic of 3D printing in bioengineering is just beginning to unfold, and it's set to transform the future of healthcare. From patient-specific surgical models to custom prosthetics and potentially even printed organs, this technology is paving the way for a new era in bioengineering. Let's explore the fascinating world of organoids and their significance in bioengineering. Organoids, if you're unfamiliar, are miniature, simplified versions of an organ produced in vitro in 3 dimensions that show realistic microanatomy. They are derived from stem cells and have the ability to self-organize in the right conditions, mimicking the complexity of the organ. This makes them incredibly valuable for research purposes. With organoids, scientists can study organ development, disease progression, and the effects of drugs on a smaller, more manageable scale. This can lead to a better understanding of how diseases develop and progress, and how different treatments can affect them. But it doesn't stop there. Bioengineering techniques have enabled the innervation of organoids with vasculature, making them more similar to human tissues. This means that organoids can now function more like the organs they're modeled after, providing a more accurate platform for testing and research. From drug testing to studying physiology, organoids are proving to be a powerful tool in bioengineering. They're helping to pave the way for more personalized medicine, better understanding of human biology, and potentially the development of lab-grown organs for transplantation. The future of bioengineering is here, and it's smaller than you might think. Let's delve into the cutting-edge world of AI, scaffolds, organoids, and synthetic cells in personalized medicine. This exciting field is revolutionizing healthcare, and it's all thanks to bioengineering. AI is not just a tool for automation, it's an intelligent system that's revolutionizing the way we design experiments. With AI, we can precisely manipulate scaffolds, organoids, and synthetically engineered cells, enabling us to create more accurate models of human organs and diseases. These models are then used to test new drugs and treatments, targeting diseases like cancer and aging at a molecular level. It's a new approach to medicine, one that focuses on the individual rather than the average. By using AI in conjunction with bioengineering techniques, we can tailor treatments to the unique biology of each patient. But it doesn't stop there. Spatial transcriptomics and next-generation sequencing are being used to identify the unique biology of individual cells. This allows us to understand diseases at a much deeper level, further enhancing the precision of our treatments. The fusion of AI and bioengineering is paving the way for a new era of personalized medicine. Treatments are no longer one-size-fits-all, they're tailored to the unique biology of each patient. It's a brave new world, and it's all thanks to the power of bioengineering. Now, let's explore the role of spatial transcriptomics and next-generation sequencing in bioengineering. Spatial transcriptomics is a revolutionary technique that allows us to visualize the expression of genes in their original spatial context, giving us a detailed map of gene activity. This technique is like having a GPS for our genes, guiding us to the specific locations where they're active and helping us understand their interactions in a more contextual way. Meanwhile, next-generation sequencing is a high-throughput method that enables us to sequence DNA and RNA much faster and cheaper than ever before. It's like having a speed reader for the human genome, allowing us to quickly identify changes in genes that could be linked to disease. Together, these techniques provide unprecedented insights into the unique biology of individual cells, paving the way for personalized medicine. By understanding the unique genetic makeup of each cell, we can tailor treatments to the specific needs of each patient. So, with spatial transcriptomics and next-generation sequencing, we're not just reading the book of life, we're beginning to understand its language. It's through this deep understanding that we can create more effective, personalized treatments, changing the face of medicine as we know it. This is the power of bioengineering, and it's only set to grow in the years to come. Let's delve into the role of AI in monitoring and analysis of spatial transcriptomics, next-generation sequencing and drug testing experiments using organoids and synthetically engineered cells. The advent of AI in bioengineering has unlocked new possibilities, with its ability to analyze vast amounts of data from spatial transcriptomics and next-generation sequencing. This computational power allows us to decipher complex genetic patterns and identify potential disease markers with unprecedented speed and accuracy. In the realm of drug testing, AI's potential is immense. It can predict the outcomes of experiments using organoids and synthetically engineered cells, providing valuable insights even before the actual tests are conducted. This predictive capability can drastically reduce the time and resources spent on ineffective treatments, paving the way for more targeted and effective therapies. But the potential of AI doesn't stop there. As we move into the future, AI is set to play an even more integral role in bioengineering. Its ability to analyze complex biological systems and predict outcomes will further enhance precision, efficiency, and personalization in healthcare. This means treatments tailored to the individual needs of patients, faster diagnoses, and a greater understanding of our own biology. With AI, we're not just shaping the future of bioengineering, we're revolutionizing healthcare as we know it. This is the power of bioengineering, and it's only set to grow in the years to come. Stay tuned as we continue to explore the exciting and ever-evolving world of bioengineering in our next scene. Let's delve deeper into the diversity of cells in diseased organs in the human body. The human body is made up of a vast array of cell types, each with its unique molecular mechanisms. These mechanisms determine how a cell functions and responds to different stimuli. Now, in the context of diseased organs, these diverse cells can behave differently, driving distinct metabolic pathways. For instance, some cells might turn rogue and start proliferating uncontrollably, leading to cancer. Others might stop functioning correctly, leading to organ failure. Understanding this diversity is crucial in bioengineering because it allows us to develop targeted treatments for various diseases. By studying how different cells respond to disease, we can design therapies that specifically target these rogue cells, leaving healthy cells untouched. This approach can lead to more effective treatments with fewer side effects. The diversity of cells in our body is a testament to the complexity of life, but it's not just a challenge to overcome, it's also an opportunity. By understanding this diversity, we can harness it to improve healthcare and treatment outcomes. This is another exciting frontier in bioengineering, and we're just beginning to scratch the surface. Stay tuned as we continue to explore these fascinating topics in our next scene. Moving forward, let's delve deeper into the diversity in molecular mechanisms and its implications on drug metabolism. Each cell in our body, as we've learned, houses unique molecular mechanisms that drive different metabolic processes. This diversity, fascinating as it is, has profound implications on how a drug is metabolized in the body. This, in turn, influences its effectiveness and potential side effects. Welcome to the world of pharmacogenomics. This fascinating field of study combines pharmacology, the science of drugs, with genomics, the study of genes and their functions. The ultimate goal? To develop effective, safe medications and doses that will be tailored to an individual's genetic makeup. But where does bioengineering come in, you ask? Well, bioengineering plays a pivotal role in personalized medicine. It helps in creating more accurate models of human organs and diseases for testing new drugs and treatments. This not only increases the efficiency of the drugs, but also reduces potential side effects. Imagine tailoring treatments to the unique biology of each patient. This is not science fiction, but a tangible reality that bioengineering is helping to shape. The future of healthcare lies in personalized medicine, and bioengineering plays a crucial role in making this a reality. Stay tuned as we continue to delve deeper into the fascinating world of bioengineering and its impact on healthcare. Let's now delve deeper into the mechanism of the CRISPR-Cas system, a revolutionary tool in bioengineering and a potential game changer in medical science, tissue engineering, and biotechnology. This system, nature's own defense mechanism, has been repurposed by scientists to edit genes with unprecedented precision. The key players in this system are the PAM sequence, the guide RNA, and the Cas protein. The PAM sequence is a short DNA sequence that the Cas protein uses as a beacon to identify its target. This is followed by the guide RNA, which is like a GPS, directing the Cas protein to the right part of the genome. Once the PAM sequence and guide RNA have directed the Cas protein to its target, the Cas endonuclease activated. This molecular scissor cuts the DNA at the precise location, allowing for specific gene editing. There are different types of Cas molecules, each with their unique features. Cas9, the commonly used, cuts DNA, while Cas12a and Cas13 target RNA. The precision of these cutting tools opens up immense possibilities. In the field of medical science, the CRISPR-Cas system shows promise in modifying cells in disease states like senescence and cancer back to their normal physiology. This could revolutionize cancer treatment and aging research, giving us novel ways to fight these conditions. In tissue engineering, the CRISPR-Cas system can be used to engineer specific tissues, can then be used in regenerative medicine. This has the potential to change the way we treat a wide range of conditions, from degenerative diseases to serious injuries. In biotechnology, the potential applications of the CRISPR-Cas system are innumerable. From increasing crop yield to creating disease-resistant livestock, possibilities are only limited by our imagination. The CRISPR-Cas system is indeed a game-changer in bioengineering, offering unprecedented precision in DNA editing. But as with all powerful tools, it also brings with it ethical questions that are still being explored. Next, we will look at how this technology is being used in real-world applications and the challenges and opportunities it presents. Let's explore how bioengineering could revolutionize the future by synthesizing artificial organs. The potential of this technology is immense, with the possibility of creating functional tissues and organs that could potentially replace the need for organ donors. Organoids and tissue engineering are at the forefront of this endeavor. Organoids, miniature organ models, are created through the process of self-organization of stem cells in a three-dimensional culture. These organoids can mimic the complexity and functionality of organs at a smaller scale. However, the goal is to scale up and create fully functional organs that can be used in transplants. The CRISPR-Cas system plays a crucial role in this process. By modifying the genetic makeup of cells using this system, scientists can create specific types of tissues. For instance, a liver cell could be genetically modified to behave like a pancreas cell, thereby creating pancreatic tissues. However, creating an organ is not just about putting together different types of tissues. The organ needs to function correctly and be compatible with the human body. This is where the understanding of human physiology comes in. Understanding how organs function in the body, how they interact with other organs, and how they respond to different stimuli is crucial in creating artificial organs that can function as well as natural ones. The synthesis of artificial organs is a promising frontier in bioengineering, potentially eliminating the need for organ donors and transforming the future of healthcare. However, this field is still in its infancy, and there are many challenges to overcome. But with the pace of advancements in bioengineering, the synthesis of artificial organs might not be as far off as we think. As we turn the page of this exciting chapter of synthetic organs, let's delve into another fascinating frontier of bioengineering: the development of nanobots in healthcare. Nanobots, minuscule robots so tiny they could navigate through our bloodstream, are no longer just the stuff of science fiction. Bioengineering plays a crucial role in designing these tiny marvels. Imagine nanobots that can travel to a specific location in the body, identify a disease at its earliest stage, and report back. Or consider nanobots that can deliver drugs directly to a tumor, minimizing side effects by avoiding healthy tissues. These are not just theoretical possibilities, but realistic goals that bioengineers around the world are striving to achieve. The potential of nanobots extends even further when we consider personalized medicine. In the future, nanobots could be designed to tailor treatments to individual needs. They could be programmed to understand a person's unique genetic makeup, identify potential health risks, and deliver preventative treatments. The fusion of bioengineering and nanotechnology is set to revolutionize healthcare, offering new possibilities for early diagnosis and personalized treatment. As with any emerging technology, there are challenges to overcome. However, with the continued advancement of bioengineering, the dream of harnessing the power of nanobots in healthcare is becoming increasingly achievable. As we step into this exciting future, one thing is clear: bioengineering is more than just a field of study. It's the key to unlocking a new era of medical innovation. As we traverse the exciting landscape of bioengineering, let's take a detour into a different yet equally fascinating application: its role in military and defense research. Picture this: Bioengineered microbes that can detect the presence of harmful chemical or biological agents, acting as early warning systems in the battlefield. Or envision super soldiers, enhanced with bioengineered capabilities to withstand extreme conditions, carry heavy loads, or heal faster from injuries. This is not a sci-fi movie plot. These are real possibilities that bioengineering could bring to modern warfare. But it's not all about weapons and warfare. Bioengineering also holds significant potential in protective measures, such as advanced body armors made from bioengineered materials that could provide unparalleled protection while being lightweight and flexible. On the medical front, bioengineered treatments could revolutionize battlefield healthcare, offering rapid wound healing or even regenerative capabilities. However, as we tread towards this future, ethical considerations and international regulations come into play. The use of bioengineering in warfare raises serious questions about the boundaries of scientific advancements and their potential misuse. While the technology could bring about groundbreaking progress in military and defense strategies, it's crucial to navigate this path with caution, considering the ethical implications and striving for a world of peace rather than warfare. As we delve into the next scene, remember that while bioengineering holds immense potential in various fields, it's our responsibility to guide its development towards the betterment of humanity rather than its detriment. As we transition from the battlefield, let's now explore a different realm of bioengineering, plant tissue culture and its potential in designing climate-resistant plants. Picture, if you will, a future where we can grow plants in artificial mediums, separate from their parent organism. This is not some far-off dream. It's called plant tissue culture, and it's happening right now. Bioengineering plays a significant role in this process, allowing us to genetically modify plants to enhance their resilience against changing climate conditions. This not only protects our biodiversity, but also ensures food security in areas most severely affected by climate changes. Imagine crops that can withstand drought, flood, or extreme temperatures, ensuring a stable food supply regardless of weather conditions. But the potential of bioengineering doesn't stop there. It might just be our ticket to space agriculture. That's right. By designing plants capable of growing in the microgravity conditions of space, we could potentially establish sustainable food sources on the Moon, Mars, and beyond. This could be the key to long-term space exploration and colonization. As we continue our journey into the fascinating world of bioengineering, let's remember the immense potential it holds not just for human health, but also for the sustainability of our planet and beyond. As we move forward, we must ensure that this technology is used responsibly and ethically for the betterment of all life on Earth and perhaps even beyond. As we venture further into the world of bioengineering, we find ourselves amidst the unseen yet powerful world of microbes. Bioengineering has the potential to harness the power of these microscopic organisms to improve soil fertility, revive climate change-affected marine resources, and even enhance human health. Imagine bioengineered microbes that can enhance soil fertility, promoting sustainable agriculture. These engineered microbes can convert non-bioavailable nutrients into forms that plants can absorb, enabling them to grow in even the most challenging environments. This could revolutionize agriculture, making it more sustainable and resilient to climate change. But the potential of bioengineering doesn't stop at the soil. It extends to our vast oceans. Bioengineering can help revive marine resources affected by climate change, preserving the rich biodiversity of our seas. By designing microbes that can absorb and sequester carbon dioxide, we can help mitigate ocean acidification, a major threat to marine life. Turning our attention back to land, let's consider our food. CRISPR-Cas engineered microbes can synthesize better fermented foods and beverages. By customizing the microbes involved in fermentation, we can enhance the nutritional content, taste, and even the safety of our food. This could lead to significant improvements in human health and well-being. As we continue to explore the vast potential of bioengineering, let's remember its power to transform not just healthcare, but also our environment and the food we consume. The invisible world of microbes, with the help of bioengineering, could hold the key to some of our biggest challenges. The future is small, and it's absolutely fascinating. As we continue our journey through the fascinating world of bioengineering, let's delve into the intricate world of metabolic pathways in the human body and their role in various diseases. Metabolic pathways represent the complex network of chemical reactions within our body that convert food into energy, and building blocks for our cells. It's like a vast interconnected highway system where each route represents a different biological process. However, when disruptions occur in these pathways, it can result in metabolic disorders. Conditions like diabetes, Hashimoto disease, hypo and hyperthyroidism, cancer, and other metabolic syndromes are all caused by these disruptions. It's as if a roadblock has appeared on our highway, stopping the normal flow of traffic and causing chaos in the system. But here's where the magic of bioengineering comes into play. Imagine being able to design therapeutically engineered cells that could cure these metabolic disorders. By understanding the metabolic pathways and the defects causing the disorder, bioengineers could potentially design cells that can correct these defects, thus curing the disorder at its root. Additionally, bioengineering can also use tissue engineering to replace defective cells in organs like the pancreas, thyroid gland, brain, skin, kidney, and heart. This isn't just a patch-up job. This is like rebuilding the entire highway, ensuring smooth traffic flow and restoring the normal functioning of the system. The future of healthcare lies in understanding and manipulating these metabolic pathways. With bioengineering, we're not just treating symptoms, we're addressing the root cause of these disorders. We're creating a future where diseases could be cured at a cellular level, giving individuals a new lease on life. As we continue to explore the potential of bioengineering, the future of healthcare looks promising indeed. As we continue our journey through the fascinating world of bioengineering, let's take a leap into the future and explore the potential of engineering biological organisms for Mars colonization. Imagine a future where we engineer unconventional nucleotides other than the familiar adenine, guanine, cytosine, thymine, and uracil. These synthetic nucleotides could be the building blocks of new synthetic life forms, designed and tailored for survival in the harshest conditions. Bioengineering could play a crucial role in creating these synthetic organisms, capable of surviving in the extreme conditions on Mars. This could pave the way for human colonization of the red planet. We could engineer bacteria to produce oxygen, break down Mars's soil into fertile ground, and even create life forms that could withstand the harsh radiation and cold of the Martian environment. The potential applications of bioengineering extend beyond our planet. Synthetic biology could enable us to create life forms that not only survive but thrive in the conditions of exoplanets in our solar system and beyond. The future of bioengineering extends beyond our planet, offering the potential to reshape life as we know it and expand our reach into the cosmos. As we look to the stars, we can see that bioengineering is not just about improving life on Earth. It's about creating new possibilities for life everywhere. Continuing on our fascinating journey through the world of bioengineering, let's delve deeper into an exciting and innovative application, using synthetic nucleotides and DNA molecules in combination with artificial intelligence for data storage. Imagine a future where we use synthetic nucleotides to encode binary data. This is the concept of DNA data storage. By using the 4 nucleotides in DNA, adenine, guanine, cytosine, and thymine, we can potentially store vast amounts of information in a microscopic space. But why DNA? The advantages are compelling. DNA data storage offers high density, longevity, and energy efficiency. A gram of DNA can store up to 215 petabytes, 215 million gigabytes, of data. Plus, unlike conventional storage methods, DNA doesn't degrade over time. It can retain information for thousands of years, making it a sustainable and long-term solution for data storage. Now, where does AI come into play? AI and neural networks play a crucial role in optimizing the encoding and decoding process. They can help to minimize errors and enhance the accuracy of data retrieval. AI algorithms can also help to compress data, further enhancing the storage capacity of DNA. When we compare DNA data storage with conventional methods like cloud storage, hard drives, SSDs, and servers, the potential of DNA data storage becomes clear. As we continue to generate vast amounts of data, bioengineering offers a promising solution to our storage woes. As we wind up this scene, let's ponder this. The future of data storage could very well be in our DNA. As we continue to push the boundaries of science and technology, we open up new possibilities that were once the stuff of science fiction. Bioengineering is not just about improving life on Earth, it's about creating new possibilities for life everywhere. As we reach the end of our journey, let's explore the experimental applications of synthetic biology. CRISPR-Cas9, and bioengineering in advancing cell and molecular biology research. Synthetic biology, a new frontier in science, aims to design and create new biological parts, devices, and systems. It's like playing with life's building blocks, but with a twist. We're not just replicating what nature has given us, we're improving upon it, creating new forms of life and biological functions. Now, let's turn our attention to CRISPR-Cas9. This revolutionary tool allows us to edit genomes with unprecedented precision. It's like having a molecular scalpel that can cut and paste DNA sequences, making it a game changer in our fight against genetic disorders. We can literally rewrite the blueprint of life, fixing genetic errors that cause diseases. But that's not all. Bioengineering is contributing to the development of innovative techniques and tools for studying cells and molecules. From creating microenvironments that mimic the conditions inside the human body to developing microfluidic devices for single-cell analysis, bioengineering is making it easier for us to study the building blocks of life in greater detail. As we conclude this scene, let's take a moment to appreciate the power of these technologies. The experimental applications of synthetic biology, CRISPR-Cas9, and bioengineering are revolutionizing cell and molecular biology research, paving the way for new discoveries and advancements in healthcare. As we continue to push the boundaries of what's possible, we're creating a future where diseases are not just managed but cured, a future where life is not just sustained but enhanced, a future shaped by bioengineering. As we teeter on the verge of a revolutionary epoch, let's delve into the collaborative influences of bioengineering, CRISPR-Cas, AGI, quantum biology, quantum computing, and virtual reality in forging transhumans. Bioengineering and CRISPR-Cas are already modifying our genetic structure to amplify our physical and mental capabilities. AGI, quantum computing, and virtual reality are granting us unparalleled tools for learning, resolution, and exploration. Quantum biology is revealing new insights about the basic structure of life and its possible effects on lifespan and health. This signifies the onset of transhumanism, and the future has never shone brighter. As we conclude our exploration, let's investigate the uses of bioengineering beyond the boundaries of medicine and biology. Bioengineering is being utilized in the preservation of the environment, with scientists designing microbes to cleanse oil spills and other pollutants, rejuvenating damaged ecosystems. In the realm of agriculture, genetic alterations are producing crops that yield more and are resilient to pests and harsh weather conditions, guaranteeing a secure food resource. In the sphere of energy generation, microbes are being designed to produce biofuels and other renewable energy forms, lowering reliance on fossil fuels. Bioengineering goes beyond enhancing human health. It's about enhancing the health of our planet, finding sustainable solutions for our future, and utilizing the power of life itself. As we delve deeper into the intriguing world of bioengineering, let's scrutinize closely how we're utilizing the power of microbes for bioremediation. We're segregating genes of interest from model organisms and transferring them to microbes. The role of the CRISPR-Cas system in ensuring correct gene integration is momentous. We're releasing genetically engineered microbes to contaminated regions for bioremediation. The application of bioengineering and the CRISPR-Cas system in creating microbes for bioremediation is a promising frontier in environmental conservation. As we delve deeper into the fascinating world of bioengineering, let's take a closer look at how we're harnessing the power of microbes for bioremediation. Using the precision of the CRISPR-Cas system, scientists are engineering bacteria, fungi, and algae to clean up contaminated areas, offering a sustainable solution to pollution. The process begins by isolating genes of interest from model organisms. These genes, when transferred to microbes, can equip them with the ability to break down harmful substances in the environment. This transfer of genetic information is made possible by vectors, which can integrate the genes of interest into the genome of the target organism. But how do we ensure these genes are integrated correctly? That's where the CRISPR-Cas system comes in. This powerful tool allows scientists to make precise nicks in the genome of the target organism, creating an opening for the vector to introduce the gene of interest. This process results in a genetically engineered microbe equipped with the ability to degrade pollutants. Once these microbes are ready, they can be deployed in areas contaminated by chemicals, oil spills, and other pollutants. There, they begin their work, breaking down harmful substances into less toxic or non-toxic compounds. In essence, these tiny organisms are nature's cleanup crew, helping to restore the balance of our ecosystems. The use of bioengineering and the CRISPR-Cas system in creating microbes for bioremediation is a promising frontier in environmental conservation. It's a testament to the power of bioengineering, not just for improving human health, but also for preserving the health of our planet. In the face of growing environmental challenges, these innovations offer a glimmer of hope for a more sustainable future. In an era where sustainable energy solutions are of utmost importance, bioengineering is playing a pivotal role in revolutionizing the production of biofuels. Harnessing the power of microbes such as yeast, algae, and E. coli, we are now able to produce biofuels in ways that are not only more efficient, but also less harmful to the environment. Let's dive into this exciting development. Bioengineered microbes hold great potential in producing biofuels that outperform traditionally produced bioethanol and are less detrimental than petroleum. By isolating and transferring specific genes of interest, we can engineer microbial strains that are highly efficient in producing biofuels on a large scale. But how is this achieved? With techniques like RNA silencing, high-throughput artificial microRNA, and the CRISPR-Cas system, scientists can identify and manipulate microbial strains to optimize their biofuel production capabilities. This precision and efficiency in gene transfer and expression is a game changer in the field of biofuel production. The impact of these advancements is far-reaching. By reducing our reliance on petroleum and other non-renewable resources, we can significantly lower our carbon footprint and make strides towards a greener and more sustainable future. In conclusion, the fusion of bioengineering and microbial technology is creating a paradigm shift in how we approach energy production. It's an exciting time as we witness the dawn of a new era in biofuel production, one that holds great promise for the future of our planet. The power of bioengineering is not just revolutionizing healthcare, but it's also paving the way for sustainable energy solutions. As we reach the end of our journey through the fascinating world of bioengineering, let's take a moment to reflect on what we've learned and the challenges that lie ahead. Bioengineering is undeniably transforming the way we approach healthcare, agriculture, energy production, and environmental management. It's giving us the tools to manipulate the very building blocks of life to create solutions that were once the stuff of science fiction. However, with great power comes great responsibility. Bioengineering, while holding immense promise, also presents potential risks and ethical considerations. The ability to manipulate genes, create synthetic life forms, and alter biological systems poses questions about the sanctity of life, the potential for misuse, and the unintended consequences that might arise. In this context, the role of international governing bodies and institutions becomes crucial. It's in their hands to regulate bioengineering research, ensuring that it's conducted ethically and responsibly, with a clear understanding of potential risks and benefits. They must strike a balance between encouraging innovation and ensuring safety, between pushing the boundaries of what's possible and respecting the natural order of things. Bioengineering holds immense potential, but it's our responsibility to guide its development towards the betterment of humanity rather than its detriment. As we continue to explore the exciting and ever-evolving world of bioengineering, let's remember to tread carefully, ethically, and responsibly, because the future of bioengineering is not just about what we can achieve, but also about what kind of world we want to create.